topic for his thesis was epigenetic regulation of the centromere activity in Drosophila, which are a type of fruit fly. In 2000 and 2004, he completed postdocs at the University of Utah and at the Stowers Institute of Medical Research in Kansas City, Missouri, where he studied chromosome imprinting in Drosophila. He was then a professor in the Department of Biology at Texas A&M until 2015, where he came to the U of A. He has been a professor here since, and he's the current vice chair in the genetics graduate interdisciplinary program. And then, assuming I've totaled it correctly, he's had about 17 graduate and 28 undergraduate students come through his lab, which he has assisted in their research and their careers. So without further ado, I ask you all to give a warm welcome to Dr. Maggert.
Okay, hi, you can hear me, right? Hi, I'm Dr. Maggert, or Keith. You can just call me Keith if it makes you more comfortable. Thank you all for having me. I'm going to talk a little bit today about the work that goes on in my lab. And I was also asked to tell you a little bit about myself and give you some advice. So if you don't care for my advice, that's fine. You can just sort of ignore me through that part. I'll get it out of the way right up front, but then I'll talk a little bit about the science that goes on in my lab. Right, so let me start. So first of all, I'm going to talk about epigenetic silencing. So how many have heard of what epigenetic silencing is? How many have heard the term? Okay, anybody want to hazard what that means? Somebody shout it out. All right, this bodes well. Okay, very good. You're right. It is silencing that's caused by non-genetic mechanisms. That is, the way the chromosome is structured, the shape that it adopts, that it folds into, the way it's packaged up, that somehow turns the gene on or off. Great job. Okay, so first of all, the people in my lab that do most of the work, these are the people in there now. Currently, I have 3 PhD students, Eric, Yule, Harvey, and Elizabeth. They're all working on their PhD. I've got 2 students that are working on their master's degree, Jacob and Sean, and 2 that are working on their bachelor's degree, Miranda and Kayla. So a lot of the work that goes on in the lab is done by these people as part of their training. I also work in the lab every day. We all sit side by side, sorting through fruit flies and doing experiments together. So as you'll see, I work primarily with Drosophila melanogaster, the fruit fly. And I use it as a model system to understand the very earliest stages of cancer development. Okay, so most of the work is done by these 2 little dudes right here. Actually, that's a lady on the left and a dude on the right. I'm sure you can all tell. And then that's me eerily hanging out in the background. So this is a fruit fly, a male fruit fly. And they've been used as a model system. That is, they've been used to study basic biological mechanisms for over 100 years, probably about 125 years now, because their biochemistry is the same, their cell biology is the same, about 80% of their genes are the same as our genes. They undergo the same developmental processes, they get cancer, they have immune function, all of those things. And so by understanding something simple like the fruit fly, we can understand something more complicated like humans. And there are some of us that dabbled in both. We do, in my lab, most of the work in fruit flies, and then we apply that to human tissue culture cells, or in some cases to human tumors that we obtain from the biobank up at the medical school, and ask whether the discoveries that we make in fruit flies are indeed true of humans. And luckily, yes. Okay, so a little bit about me. My very first job was feeding penguins. So I was born into a biology family. My dad was a physiologist that used to winter over in Antarctica. In fact, all the penguins at the San Diego SeaWorld were brought back from Antarctica by my dad. At least their ancestors were, because now we're in 2nd or 3rd generation since. So he used to winter over and collect these emperor penguins, bring them back, and then do experiments on them at Scripps Research Institution, or Scripps Institution of Oceanography, rather, in San Diego, and I would go there on the weekends and feed them. But I worked on other organisms. I worked on tarantulas. Any guess why somebody would want to work on a tarantula? It's a great model system for thermal regulation because they're cold-blooded, but they have hair, right? So normally we think of hair as insulative, but these things are cold-blooded, so they don't want to insulate. So what's the deal with tarantulas? I worked on that when I was in high school and at the beginning of college. I also worked on carrots. Carrots are surprisingly sweet fruit, roots. And I worked on the biochemistry of how that much sugar was pumped into the vacuoles in carrots to make them sweet. So the actual work was not very interesting, if I do say so myself, but boy, oh boy, the carrot cake recipe that I got out of that lab kicks ass. Okay, I worked on yeast a little bit. That's pretty boring. Other people work on that too. And then the last thing I worked on was this weird organism called Oikopleura. It's a larval tunicate, so it swims around in the ocean. It's basically just a head with a tail, and it just swims around backwards, running into stuff. And the way it feeds, because its mouth is in the front, but it's always moving backwards, is it's got this huge, enormous snot tent that it inflates around itself. So it's got these secretory cells on the top of its head, and it secretes this mucousy snot up there. Well, it's got a little top hat. And then it starts to wiggle water into it, and it inflates, and then it jumps inside, and it's like this little motorboat that goes driving backwards through the water. And here you can see it, and it's adorned by these enormous filters. So as it goes backwards, all the plankton goes into these filters and is fed directly into its mouth. So how in the world those cells on its head know how to secrete a snot tent that ultimately will form such an intricate structure is a fabulous mystery in biology. So I tried for a while to work on that, and it was well beyond my capabilities. So I ended up with fruit flies, and I've been working on chromosome biology ever since. I was asked to give you all some advice on how to be a scientist, and I don't know if this is going to ring true or bullshit or whatever, but it's what worked for me, and it's in my observation what works for other people. And this is the difference between a scientist and what I consider to be a great scientist. And it really just boils down to one thing: read. So there have been hundreds of years of work done on whatever project it is that you think you may be working on in biology, and hundreds, if not thousands, of papers and opinions and ideas and experiments that have all fed into the current view. Read that stuff. You will be famous, you will be fantastic, you will be a clever and amazing scientist if you know what people did 20 years ago, 30 years ago, 40 years ago. So read the old stuff, read the new stuff, of course, and most importantly, read stuff that you think has no bearing on you. Because sometimes the ideas that we get that are most foundational for our progress come from things that we didn't even think were relevant. Ask questions, and I mean that both sort of metaphorically as well as realistically. If you want to ask me questions, just raise your hand or shout out, and I will be happy to answer them. And of course, make personal relationships. So one of the great things about the UBERP is that you're making personal relationships with other people in the lab, with these principal investigators, with professors, and all of that stuff is going to open doors for you in the future. So congratulations to all of you for being involved in lab research so early on in your career. That's going to make all the difference. So just quickly, how many of you want to be professors? All right, not bad, so we don't make it look terrible. What do other people want to be? Clinicians? Lawyers? Be in biotech? Be rich? Be happy? Oh, okay. All right. And I urge you to find the right place for you. That is a place that is happy, that stimulates your curiosity, that gives you joy to go every day. Because frankly, and I think Jacob and Miranda will agree with this, the job can be really fucking drudgery, really depressing. Right? As I say, the best day in a scientist's life is the day that he fails to disprove his own hypothesis. And that's kind of a miserable way to live. Like, oh, today I'm going to disprove my hypothesis. Failed. Yay, success. And it just goes again and again and again. And some of it can be pretty technically challenging. And so you need to have a motivation and a joy in order to make it through. So now I'm going to talk a little bit about the science. Is there any questions about that, about me, about the advice? Pretty straightforward stuff. Okay, I'm going to sip some water. You're going to watch. Very good. Okay, so how many have heard of the word heterochromatin? Okay, so everybody's doing these little half hand raises. I'm not going to call on you, I swear to God. How many have heard of heterochromatin? All right, that's a 3/4 hand raise. You guys are tough. Okay, so heterochromatin is the structure of a chromosome that is adopted by cells that is an extremely stable structure. So of course, all of the cells in our body, more or less, have the same genomes. They all have the same sets of genes, but not every cell is doing the exact same thing, right? A neuron is different than a kidney cell. The kidney cell is different than a muscle cell. And so what discriminates how those cells behave is which genes are being activated and which genes are being repressed. And I care more about repression because I've always been sort of a dour, depressive sort of guy. Activation, that's for the happy people. I'm going to take repression. And the structure that genes adopt when they repress things is called heterochromatin. It's called that way because it's a different color, heterochroma, when you look at it in a cell. And I've got some pictures of that later. Heterochromatin is used to shut off genes that are not currently being used or genes that are universally toxic. And I'm going to talk about some of these universally toxic genes in a little bit. When heterochromatin is functioning, the cell is functioning properly. When heterochromatin breaks down and fails to function, then genes that should normally be off can inappropriately be turned on, and that can lead to all sorts of problems. One such problem is cancer. When heterochromatin fails to shut off genes that are involved in the cell cycle, for instance, the cell division, then you get out of control cell division, and that is by definition cancer. So normally heterochromatin gives rise to some sort of variation. Sometimes it's functioning, sometimes it's not, because nothing is perfect in this world. And amongst the trillions of cells in all of our bodies, not every one of them is going to get it right every single time. So the phenotype that arises when heterochromatin is dysfunctional and it's not appropriate is called position effect variation. And I'll show you pictures of it and explain to you where it gets that name. Position effect variation, or just PEV, is an epigenetic phenomenon. That is, two cells that are behaving differently in terms of position effect variation are no different from each other genetically. Their DNA sequences are identical, and yet they have very different phenotypes. So that's epigenetic. It is under developmental control, so the function of heterochromatin changes throughout the life of an organism. And we, in my lab, developed a way to monitor it in live cells, so we can understand how heterochromatin functions and how it contributes to shutting off toxic genes that can otherwise give rise to cancer. Okay, so variation, of course, is normally something that's very good. We think about variation happening within populations and whether we're talking about directional selection, stabilizing, or disruptive selection. Variation between different organisms is what evolution acts on. So if evolution wants something which is bigger, it will select for the bigger things on the variation axis. If it wants something in the middle or something on either side but not the middle, these are the types of ecological variation that give rise to evolutionary trends over long amounts of time. That's on a population scale, but the same thing is happening in cells and at genes. There's variation from cell to cell as to how genes are functioning and how effectively those genes function. So if there's too little variation, it creates vulnerability. If some insult were to happen to an organism or to an ecosystem where variation is reduced very low, the entire ecosystem, the entire organism could be wiped out. Think about the Irish potato famine. Because it was a monoculture, one virus or one smut actually was able to wipe out the entire population. But in excess, too much variation can result in disease. And that's the thing that I study in Drosophila. Okay, So, like most things in the 21st century, it all comes back to Barbara McClintock. For my money, the most brilliant scientist of the 20th century, and yes, I include Einstein. She would have kicked Einstein's ass. She got her Nobel Prize in 1984 for proposing that genomes are living organisms, or they can be likened to living organisms. That a genome is able to, should I not be moving? Oh, you're just getting that. Okay, I'm sorry, I'm not drawing attention to you. Okay, so she proposed that, what did she propose? I've forgotten now. She proposed that genomes sense the environment and can adapt to the environment so that insults that come from stress or starvation or dehydration or whatever can be adopted into the genome, and that genome can change itself so that the organism is better to withstand it, and that organism's offspring are better able to withstand it. So this sounds very Lamarckian. And no doubt Darwin was right and Lamarck was wrong. However, Barbara McClintock was onto something, and she had a mechanism to understand how genomes can adjust themselves. And as she said in the white, the genome is a highly sensitive organ that senses unusual and unexpected events and it responds to them. We know nothing about how the cell senses danger and instigates responses. And her life's work was to understand how the genome senses changes and how it adapts to them. And it turns out that that process, which is key for evolution, how genomes can change over evolutionary time, leads as a consequence to diseases like cancer. So they are the same process. She discovered the importance for evolution, but it turns out that the exact same process that we now study in Drosophila can lead to disease. And that is all mediated by heterochromatin. Okay, so heterochromatin, here it is. This is an electron microscope photograph of a cell. You can see the gray stuff, that is the nucleus right here. And the black stuff within the nucleus, that is densely staining DNA. That is the DNA that is tightly condensed and inaccessible to transcription factors, to RNA polymerase, etc. So those are the genes that are off. They actually have a physical manifestation in the cell as these dark blocks of heterochromatin. This thing in the middle, that's the nucleus. I'm not going to talk about that. And then the grayer stuff around the outside, those are the active genes. And so the genes will segregate themselves within the nucleus as they're active or inactive, that is, whether they are heterochromatic or euchromatic. This is what it looks like in a metaphase spread. So each of these is a chromosome. Here's the two sister chromatids together. There's two more sister chromatids here, two here, and two here. And then these are the two sister chromatids for the small fourth chromosome. And you can see that some areas are brighter than others. Here, the Y chromosome is almost entirely bright. That is heterochromatin. So we can see the differences between the bright areas, the heterochromatin, and the dark areas, the euchromatin, where all of the genes are. So what would happen if I were to take a gene out of the euchromatin and put it into the heterochromatin? It would be shut off, and it's shut off in a very particular way. So here's a teeny little picture. I don't know why there. Okay. So this is a picture that I took about a year ago, and this is a picture that comes from a publication in 1931. So that's what I mean, that this mystery of how genes turn off has been around for about 100 years. So what you can see, oh, shit. Well, that was disconcerting. Is that going to happen again?
It already happened before. It barely blips off, and then it'll turn back on.
Okay. All right, well, there goes my flow. Anyway, so what you see is the eye of a fruit fly is normally red, but there are white patches in the eye where there's no pigmentation in the eye, and that's because the gene that makes the red pigment is shut off in those areas. So we can look right here. Okay.
It's back.
We can look, I'm going to get off this slide right now. So we can look at those patches and see that there are cells in which the gene is off, that the heterochromatin is functioning, and cells in which the gene is on and the heterochromatin is not functioning, because it's heterochromatin which shuts that off. This is a picture of the human genome. There's a long chromosome, and then this is the region that's blown up. And what you can see here is some gene. I don't even know what gene it is. It doesn't matter. But you can see that the actual coding region is demarcated by these little lines. So most of the gene in the human genome is not coding region. It's intron. And that intron, what is it made out of? Well, it's made out of these repetitive sequences called transposable elements. So how many have heard of transposable elements? Okay, so this is the toxic gene set that I'm referring to. These are the genes that normally cause huge amounts of damage to the genome that Barbara McClintock realized could rearrange the genome that are important for evolution but ultimately lead to disease. So you can see in these three different traces down below, this middle one, these are healthy cells, and each of these peaks here indicates the location of heterochromatin. You can see that the heterochromatin matches up with the location of these repetitive sequences. It is heterochromatin which is shutting off the transposable elements and keeping them from hopping all over the genome. In two different cancer lines, this is an ovarian cancer and this is a bone cancer. In both cases, the heterochromatin is missing. The transposable elements are free to mobilize and cause damage to the genome. Oops. And there's not just a couple of transposable elements. If I just blow up this region of a gene, you can see here each of these little blue rectangles on the DNA sequence is another transposable element. So it's no joke, your genome is about 3/4 transposable element, which is a huge amount of potentially damaging DNA that is all being controlled by heterochromatin. Okay, so this is just to remind you what transposable elements are. There's two different kinds. There's DNA-based ones, which are little DNA sequences which can cut themselves out of the genome and move to a different location of the genome. And then there's RNA-based ones which don't cut themselves out, they just copy themselves and move to a new area of the genome. So throughout the course of your life, maybe you're getting 1,000 mutations in each of your cells that are caused by mobilization of a transposable element. You can imagine that if one of those things hops into a gene that's necessary for cell cycle control, that would be a precancerous condition. Okay, so DNA elements, transposable elements are extremely mutagenic, and you can read through the list of ways that they're mutagenic. But the upshot is heterochromatin represses transposable elements. Transposable elements cause mutations that lead to disease. So that's why I study heterochromatin. Questions about that? Okay, so how do I study it? With the fruit fly eye. So the white gene, that is the gene that makes the red pigment in the fruit fly eye, is out at the tip of the X chromosome. And a gene which is fully functional will produce an eye which is bright red. If you've ever seen a fruit fly in the supermarket, you can see as they fly by, they've got the red eyes. A mutant whose gene has been disrupted is a white-eye mutant. That's why we call it white, because the mutant causes the eyes to become white, therefore it's the white gene. But if I just take that gene and I put it into heterochromatin, then I get that patchy, some are red, some are white. So it's important here to underscore that every cell in that eye is the same, and yet some of them, the heterochromatin is functioning and this gene is off, and in other ones, the heterochromatin is not functioning and the gene is on. So just by moving the gene into the heterochromatin, we create a metastable state where half the time heterochromatin is functioning and half the time it's not. And so we can understand the difference between those. If we want to bolster heterochromatin's function, we can look for flies whose eyes are whiter. And if we want to compromise heterochromatin function, we can look for flies whose eyes are redder. And that was the source of a bunch of experiments looking for things that turn this eye, where half are functioning and half are non-functioning in terms of heterochromatin, into a fly which was totally red or totally white. So again, the location of the gene in heterochromatin is the same between these three. What I've done is just mutated some other gene in the genome here or some other gene in the genome here. Right? So I removed pretty much all of heterochromatin function in this condition, and in this condition I've enhanced heterochromatin's function. But note, there's still a little red dot right there. there's still one cell where heterochromatin is not functioning. So in our view, that is the cell right there that will go on and form cancer. That is the cell in which hypermutation is occurring prior to the development of a cancer-like condition. That's why we can study fruit flies, because we can do this thousands of times in the lab, isolate those cells, literally just cut them out of the heads of flies, put them into a tube and study them. Now this is turning off. Will this come back on inexplicably as well?
I would imagine not.
Okay, I'll go rock star. Hello. Okay. So that allows us to define the components of heterochromatin, looking for things that are disrupted in those conditions that lead to all red eyes, then reveal the components of heterochromatin, and those end up being a bunch of proteins that we've now characterized. HP1, H3K9 methyltransferase, DNA methyltransferases, etc. So I'm not going to go to the biochemistry. I'm just going to talk a little bit more about the phenomenology of this. Okay. So heterochromatin itself, as you've seen, although maybe it didn't stand out at the time, is an epigenetic memory. This was an important discovery around 1950 when people realized what this meant. So here's a picture of an eye, the same picture you've seen before, that's undergoing position effect variegation. That is the part red, part white phenotype. And you can see that there's a whole patch of cells that are right next to each other that are all red. And here's another big patch of cells that are all red. And then here's a patch of cells that are all right next to each other that are all white. Well, why are they right next to each other? What does that imply about what happened to turn those cells red or white? Because each of those cells was derived from a precursor cell, from a mother cell, and each of those cells was derived from a mother cell. So the fact that the cells with the same color are next to each other means that back in time, multiple generations before, a single cell decided to either be on or off in terms of heterochromatin, and then through the next cell division cycle, that was remembered. And then through the next one it was remembered, and then through the next one it was remembered. So there was a memory of this epigenetic information. Remember, this is not a genetic change. There's no mutation in here, so it can't just be DNA polymerase which is propagating these mutations. There's something else about the chromatin structure which is transmitted from generation to generation. So this is epigenetic memory. What happens to the great-great-great-great-grandmother cell is still seen in the daughter cells. So either this gene is on or that gene is off based on how badly the heterochromatin, the sort of thicker bars here, repress it. And then through cell division, that is retained. Oops, too many animations. That is retained. And so you either get these cells where the red gene is off and you get a white patch, or the red gene is on and you get a nice red patch of cells. So we study heterochromatin in part because of this epigenetic memory. We'd like to understand how it is that the memory is maintained through each cell division. Okay. Questions about any of that? Alrighty, next. So, you want to understand Maggert. I have my own segue. So one thing that we noticed while we were doing this work is that there's different kinds of patterns of position effect variegation. So I've shown you this eye, and you can see that there's these big patches of contiguous cells. Fine, we discussed that. But there's another kind too, which looks more salt and pepper, where it looks like every individual cell is making its own decision. There is no epigenetic memory. It is the same process. It's still mediated by the same genes that silence things. It's still mediated by heterochromatin, but it doesn't seem to have the same memory. So what's the difference between those? This was observed also at the beginning of the 1900s when they first discovered position effect variegation, and there's still no adequate explanation for how. The initial explanation was these are decisions that are made early in development, and these are decisions that are made late in development. That is, if a gene is going to be silenced by heterochromatin early, it will be remembered, and the pattern will look like that. And if it is going to be made late in development, then it will look like that. Boy, the timing on this is awesome. Okay, so in other words, early in development, I can make a decision, and that would lead to this patches kind of position back variegation, or I can make it late, and it will lead to these salt and pepper kind of variegation. And we had problems with this model, and so one of the first things that my lab did was to try and address whether that was true. And what we were able to do was disprove this idea by showing that all of these decisions are made at the same time in development, and in fact, they're made at the very earliest time in development. So let me show you how we did that. We looked at fruit flies that were siblings of each other. That is, we took large populations of fruit flies that were all undergoing position effect variegation, and like I said, that itself leads to a population variance. So within there, we can find flies where there's hardly any silencing, and we can find flies where there's a ton of silencing. These are genetically identical siblings to each other, and so this is sort of the extent of the variation that we see in position effect variation. And we just simply looked at their other eyes. We looked from the left eye to the right eye and the left eye to the right eye. So if silencing is determined randomly at each gene, then there's no reason to think that the left eye and the right eye will be correlated with each other. But if silencing is controlled by something which is intrinsic to the animal, that is something that affects its entire body, then the left eye and the right eye should obviously be correlated. So we did that correlation analysis. We looked at hundreds of left eyes and hundreds of right eyes and correlated them to each other. And this is the logic behind it. If we start with a single cell at fertilization, and then that goes through a bunch of rounds of division to 2 cells, to 4 cells, to 8, to 16, 32, to 64 cells, you can imagine that this is the entire organism. And anything on the left side and the right side will only be correlated to each other if it was correlated to a common cell at some point early. So if cells here and cells here are correlated to each other, that means that they had a common ancestor that also shared that correlation. Whereas if cells here and here are correlated with each other, that means that those two patches of cells are related to each other. So something along the midline is less informative, but what we're looking at is whether these cells and these cells are correlated to each other. And we know that we can sort of backtrack that these cells and these cells are related earlier on through their development, and earlier, etc. But if I look at the left eye and the right eye, then I have to ask, at what point were those two cells the same cell? And the answer is the very first cell division. So if there's a correlation between the left eye and the right eye, then that means that the variation that contributes to the correlation between the left eye and the right eye had to exist at the time of fertilization. Did that make sense? Okay. So we did that analysis. It was a long, long week of counting flies. So what you see here in the circles is the data set if I looked at every eye individually. If I looked at all of the eyes for which there is 25% of the eye is covered with pigmentation, 50%, 75, 100, or 125 just means extra, extra bright, but I had to code it some way. So the white circles indicate if the eyes were taken at random. And the circles are the actual data, the orange dots here. So you can see that there is a enrichment along the one-to-one correlation, indicating that the left eye and the right eye are indeed correlated with each other statistically. We can do that with multiple different variegating lines, and we can do that in multiple different conditions with at higher temperature where the variegation is reduced, at lower temperature where the variegation is enhanced, and with chemical things that modify it. So we can say that without a doubt, the left eye and the right eye are correlated to each other. Therefore, what is contributing to the variation must have happened early in fertilization and not later in life. Okay. So we can look at other kinds of correlation. We can look at green fluorescent protein and look on left and right at different tissues as the organism is living and crawling around in the food. And of course, left and right is correlated in the eye. It's also correlated throughout the body, throughout development with green fluorescent protein. We can look and see whether the green fluorescent protein variegation is correlated with the eye color. And in fact, that is too. So two totally different organs within the organism are also correlated, which also shows that it maps back to the very first cell division. And we can look temporally, where we can take the green fluorescent protein variegation early on in development, and then take the ones that are highly expressed or low expressed, move them to different vials, and then measure their eyes after they grow up. And in fact, what we see is those flies that as larvae had a lot of green fluorescence indeed have a lot of red eye pigmentation as well. So every way that we do this, we can show that an individual is different from a different individual based on something intrinsic in its body, not randomly based on gene activity. So what that means then is that the variation maps to the point at which the left and the right sides were the same, and that is heterotely fertilization. So we can't say that variegation happens early in development or late in development to give those patterns. Rather, everything has to happen early at the same time. Does that make sense? So how in the world do we explain those patterns if it's not early versus late? That was the next challenge. So everything happens early. Silencing is established there. And so the question is, how do I get those two patterns? So we racked our brains and we thought, well, maybe it's not an issue of the time at which silencing is established, but rather it's an issue of how well it's established. And so maybe these patches are relatively stable with infrequent switching, and these are frequently switching. And so these are constantly turning on and off throughout development. So in order to address that, I have until 4:15, right? I'm going to skip through our mathematical modeling, which convinced us that we were probably right, and go straight then to the test for it. So what we were looking for in order to support our hypothesis was conditions where we could see switching occurring throughout development. The existing model was that silencing was laid down early or late in development and was from that point on stable, so we should never have seen switching throughout development. But we proposed that there was switching, and so we had to develop a way to look for switches, for a gene turning on or a gene turning off in a piece of tissue through development. So I'm not going to go through the mechanism of how we did it, but here are the data for it. This is an organ. This will actually become the wing and the back of the fruit fly. And what you see are two different fluorescent proteins that are being expressed at the same time. Green fluorescent protein is showing historical silencing. If the gene has ever been silenced in the past, in the development of that tissue, it will be green. It's a way of permanently tagging something as silenced. Whether that silencing maintained or not doesn't matter. If it was ever silenced, it will be green and will be permanently tagged as green. So will all of the ancestors or the descendant cells. And the red ones, those are the ones that are currently silenced. And so I can look at the four different colors. I can look at green, not red, and say, well, it used to be silenced, but it's not anymore. I can look at the spectral overlap, red and green, that is yellow, and say it used to be silenced and it's still silenced. I can look at the black cells that are non-fluorescent and say it was never silenced and it's not even silenced now. Or I can look at the red ones and say it was never silenced in the past, but now it is. Those are the most important ones. Those are the ones where silencing just happened. So if I can see red cells in amongst all of this tissue, then that says that silencing can come and go throughout development. And so that's what we see. So four things stood out at us. First of all, silencing is not established ever in some cells. Those are the ones that are black, right? Those are the ones that have never had silencing. Silencing can be gained and then lost again. Those are the green cells, right? So those are the cells that have been silenced at some point in the past but are no longer silenced right now. That is heterochromatin was functioning, and now it's no longer functioning. So the green cells itself tell us half the story. The most important one, which you can barely see here, but you can see on these spectral separations. So these are the green ones presented as black on white. Here are the red ones presented as black and white. And look at this cluster of cells right here. These are all red, and they don't exist down here in green. That patch of cells, all right next to each other, just acquired silencing. So we have evidence that silencing can be gained. We have evidence that silencing can be lost throughout development of living tissues. So this disproves the idea of early versus late and supports our idea that everything is set at the same time and it's either stable or not stable. And look that they're all right next to each other. Isn't that convenient? We noticed that as well a couple of times, a couple of places. Here's another image of it. Whenever we see these cells acquiring silencing, they all seem to be right next to each other. Not right next to each other, but all within the same area. So we're not quite sure what that means, and that's sort of what my lab is studying right now. Why is it that some cells tend to gain or lose silencing, but also why is it that some domains of tissues tend to have a lot of cells that gain or lose silencing? We think that that may have something to do with cancer onset, at least that's the hypothesis that we're pursuing. So here's the conceptual model. As I finish up, silencing is established, but it fluctuates. It goes up and down and up and down. And silencing is sufficient to keep transposable elements from damaging the genome, provided it stays above some sort of threshold. And here, that's a pretty good cell. It's fluctuating, but it's always above that threshold. But there are other types of cells that sometimes will dip below, or maybe other cells where it's even more extreme. And so in those cells, each of these little periods, even though the silencing recovers, during this period, during the period where the amount of silencing is below the threshold, those are times that transposable elements are damaging the genome, causing hypermutation that can lead to disease. So even though the situation corrects itself, the damage that happened during that brief period of time is perduring. So those are the danger points. And there are obviously conditions, some genetic mutations or other environmental conditions that just reduce the amount of silencing overall, and those are people that suffer high frequencies of cancer development. And so what we'd like to do is to create therapies or treatments that push the silencing higher in order to protect the genome. Okay. So, in summary, silencing is established early around the time of fertilization or around the time of egg laying. And that seems to be the case with humans. When these sorts of experiments have been reproduced in humans, it does seem to be correlation across the entire organism and not just at every individual gene which is undergoing variation. Second of all, silencing is incomplete in early cells, and we think that that incomplete silencing is what's leading to precancerous conditions. Third, that these losses and gains of silencing are constantly ongoing. And I'm not going to talk about some mutational analysis, but we can, because of the power of fruit flies, we can expose these things to mutations or the drugs and do sort of probatory experiments to see how we can, and if we can, adjust the silencing rates. And I will finish with this. Studies of disease, to me, are driven primarily by fascinating questions. I don't go about this thinking about cancer every day. What I think about is how fucking cool position effect variegation is and how interesting those patterns are. I mean, the idea that it is relevant to human cancer is always there in my mind, but what really drives me is the sort of interesting, specific question that I'm working on. And frankly, the random patterns of variegation in the fruit fly eye is what keeps me looking in the microscope. Disease is always a consideration, but oftentimes it's hard to explain. You've sat here for 45 minutes while I've showed you slides. You can imagine how difficult it is when somebody asks me, so what do you do for a living? How do you help try and prevent cancer? Well, it's a complicated story. Okay, so of course I hope that my work contributes to understanding how cancers begin. I hope that my work will lead to some sort of prevention or detection mechanism But I'm also fully aware that we're a part of a community of scientists, and I may not be the one that makes the final step. Maybe somebody will use my data, somebody will use my ideas, maybe it'll be one of my students, I don't know. But I think that understanding that model system biology is important and part of the step towards greater things, including human disease prevention, is an important consideration. And that is it. Thank you very much. I can take questions if there are any.
I have one.
Yeah.
I believe a couple slides back you were showing like the patches of like you were showing like the red patch and the white patch, and I saw one thing that interested me. Between the patches there was like a bridge of white between the two reds and the white. I don't know. Was that like was that interesting to you? I guess because like you were saying that line that you were interested in like the.
It's all interesting to me. You mean this line right here?
Yeah.
Yeah.
That was that I interested you when you were researching or?
Yeah, so yes, it is interesting and I know why it happens. So when the cells of the eye first are set aside, there's only like 8 or 9 cells that will give rise to the entire eye, right? So that original unlogin is set aside, and then each of those cells divides a couple of times to give you the final 800 cells of the eye. So how do you get from 8 cells to 800 cells? You go through all those cell divisions, and then how do those cells sort of orient themselves and move around? Well, they move around in kind of stereotyped ways, and one of those cells will give rise to a long sheet of cells that is the midline of the eye. So the fact that this is white right here means that the progenitor from that silenced its gene, and that all of its descendants were also silenced. And so if I were to look at 1,000 different eyes, 990 of them will either have a red line that goes across there or a white line that goes across there. But yeah, very good eye. Yeah.
So I don't know if this is exactly in your area of expertise, but with the discussions going around about designer babies and like CRISPR using that on like bacteria and things like that, so gene splicing, removing, adding genes. Do you, in your research, feel like there will be any sort of like consequences that maybe we aren't thinking of, or any sort of maybe way that the heterochromatin won't form correctly if we splice out those certain genes or if we add certain genes?
Yeah, so one caution that I would give you is that what Barbara McClintock first discovered was when you stress a cell, the transposable elements move around. And since then, there's been a lot of work that shows exactly why. Because when a cell is in a stress condition, when it becomes quiescent, right? So when you stress a cell, it stops its cell cycle. And one of the things that happens during that pause, especially when you cause DNA damage, which CRISPR does, is the heterochromatin falls apart. And so we know that when you stress a cell, it becomes hypermutated for a short amount of time. And this is a consideration that most people don't think of when they talk about doing those sorts of experiments, right? That when you do cause a mutation or when you treat your cells badly, in general, you are creating mobilization events of transposable elements. So, yeah. Yeah.
Super interesting topic.
Thank you.
I was curious to know if, in between all these different, so I'm very fascinated by the microscopy that you're showing of the swim assay. Are there any different, have you guys done any cell sorting and looked at any either proteomics or metabolomics to know what specifically is it that causes that specific phenotype to happen?
Right, yeah, so absolutely that is on the list of things to do next. Because we have color labeled these cells into the 4 different categories, we can split apart the tissues and collect all those cells in which silencing just happened, or in which silencing happened and then fell apart. And we would love to know the differences in what the proteins are, what the proteins are doing, if there are any structural changes. You know, our guess is the green cells will have more older transposable element mobilizations, and the red ones will have fewer. But those are hypotheses to be tested. But yes, exactly, we intend to do that. Yeah.
Have you done any testing to see how this is altered in like
Yeah, absolutely. We can alter the patterns of the red and the green and the yellow and the black by dietary manipulation, by treating with drugs, by treating with mutations. And so one thing that sort of surprised us, I mean, it didn't surprise us, I think it's an important note, but we fully expected it, is a lot of anti-cancer therapeutics that affect chromatin structure cause a general de-repression of transposable elements. And so, like all of these things, there's good and bad that comes with it. They're in clinical trials now, but it seems clear to us that they're going to cause damage to the genome as well. And so whether that's a net positive for a patient or not is going to have to be evaluated. But yeah, certainly we are surprised even by the sort of boring everyday stimuli that can dramatically affect the amount of silencing that there is. Yeah.
So you're looking at the behaviors other than just like physical phenotypes, you look at how prior after.
You know, we haven't, but that's a great idea because one of the places where we see the most dynamism is in the brain. So we sort of presume that there's going to be changes in patterns of neural function, but we just haven't looked at that. Yeah, but that's a great idea. Yeah. Right, because most evolution is by transposable elements. Yeah, most variation that arises in populations is caused by mobilizations of transposable elements. All right, well, thank you all very much. I really appreciate your attention.